Good afternoon and welcome to the second Van Andel Institute Public Lecture Series, a focus on pancreatic cancer, from foundations of science to early detection. My name is Brett Holloman. I'm the Chief Development Officer here at Van Andel Institute, and it's my privilege to welcome you today to the Institute and join us in highlighting both the Institute's work in basic research as well as pancreatic cancer research. Today we have the privilege of hearing from two of our research faculty, Dr. Bart Williams and Dr. Brian Hobb. Dr. Williams received his PhD in biology from Massachusetts Institute of Technology in 1996. For 3 years, he was a postdoctoral fellow at the National Institutes of Health in the laboratory of Harold Varmus, who was the former director of the National Institutes of Health. Dr. Williams joined Van Andel Institute as a scientific investigator in July of 1999. He is now a professor and the director of the Center for Cancer and Cell Biology. Dr. Williams is also one of the very first principal investigators to be recruited to Van Andel Institute when it was founded in 1996. Our second speaker today is Dr. Brian Hobb. Dr. Hobb obtained his PhD in chemistry from the University of California at Berkeley in 1998. He then served as a postdoctoral fellow in the Department of Biochemistry at Stanford University. Dr. Hobb joined Van Andel Institute as a special program investigator in May of 2000. He became scientific investigator in 2004 and was promoted to senior scientific investigator in 2007. Dr. Hobb also serves as an assistant dean of Van Andel Institute Graduate School and as an adjunct professor in the Department of Biochemistry and Molecular Biology and the genetics program at Michigan State University. We are fortunate to have both of these gentlemen with us today. Dr. Bart Williams will be our first speaker, who will introduce us to the role of basic research, prior to Dr. Hobb, who will then share his translational research and perspective on pancreatic cancer. Please join me in welcoming Dr. Bart Williams.
So good afternoon. Nice to see a nice turnout here today. And as Brett said, it's sort of my role to set the foundation part of this lecture. That is, how does basic research influence translation into clinical applications? And I hope to provide and convey some of that sense to you today. So I'd like to start these talks, especially in this type of setting, with this picture, because it kind of provides a context for how long or how far we've come. So actually you can kind of see on the screen here, this is the way the Van Andel Institute looks right now. This is Crescent Street going up the side here. This is Crescent Street here. So this is the location where the Institute is now. 20 years ago, this was a parking lot, and I believe 25 years ago at this point, and these were dorms for nursing students and residents at Spectrum next door. So obviously we've come a long way in 20 to 25 years. Just presenting this because it's important to sort of note that both Brian and I do work to try to translate our work into applications, and so my disclosures are shown here. That is, companies that I work with to try to, again, advance our knowledge that we gain in the basic research setting into human clinical application. So the overview of what I'll present today, give you a little bit of a sense about who works on a research laboratory. Many of you have some sense of that, but just sort of talk about who's in my laboratory and very briefly what their backgrounds are. And then talk about what basic research is. So we often hear terms, or people talk about terms like basic research, clinical research, translational research. What do those really mean, and what does that mean for us here? And then I work on something called Wnt signaling, WNT. It's a funny name, and you often get the question of, well, how do you decide what to work on? So my lab actually does do some work in pancreatic cancer, not to the same extent or for the same length of time as Brian's laboratory has, but we do projects that are related to that. It's because of what we work on. This funny named pathway called WNT is relevant for a lot of different things, and I'll talk about how we chose what to work on in our lab. So the members of my laboratory are shown here. Typically, a research laboratory is comprised of individuals that may have gone and got their bachelor's degrees and then decided that they just want to really focus on working directly in a lab, working at the lab bench. So I have 3 of those types of individuals in my laboratory. And then we have people that are in our graduate program. Katie is a graduate student in the Van Andel Institute graduate program, so she's working towards getting her PhD. And then there are individuals that go and get their PhDs and want to receive more training before they perhaps run their own laboratory or go work for a pharmaceutical company, a biotech company, teach in a small college. And those 3 individuals are shown here. Megan is sort of a special case. She's both a dentist and a PhD, so she has background that's sort of interesting for many of the things we work on in terms of applications to the bone and the skeleton. So overall, really the role of biomedical research, trying to sum that up on this slide, is that we try to understand how cells, tissues, and organs normally work. That's really one way you could describe basic research. And then we try to understand the causes of diseases to allow for prevention or detection, which Brian is going to talk about, and then develop more cost-effective ways of treating disease as well. So the types of research, I wanted to go into this in a little bit more detail. So really another way to talk about basic research is it's just motivated by curiosity to understand the unknown or improve our understanding of how things work. And this is really what a lot of research is focused on in the Institute here and in many universities throughout the country. And this often has unintended benefits. Really, you're trying to understand something that's completely unknown and you don't even know what it may be applicable to when you do understand it. So it could be as simple as just how do cells divide? How does one cell become two? And trying to understand that in great detail. How do cells move? How do they attach? Many of the therapies that are in use today in the clinic for many types of cancer are just based on things like that were understood initially in yeast or fruit flies or worms or very simple organisms that then we could, once we understood those processes, we could then translate that knowledge into clinical application after many translational type research projects to move towards that. I'll go to clinical research sort of next because translational is really the interface between them. Clinical research, and this is the definition that's provided by the National Institutes of Health, is research in which people or data or samples from people are studied to understand health and disease. So these are obviously driven by medical doctors, MDs, who have research training working with people like us to try to understand things and move things forward in the application of knowledge. And then translational research is really the interface between them. A lot of times people don't really have a great understanding of what translational research is, or at least a great definition. It's really trying to apply what is seen in a basic research laboratory to a clinical application. And that's actually the most difficult part of this in many cases, is really trying to move the basic research towards the clinic. And Brian will talk about his work that really is translational in nature, looking at detection and trying to then build that into a clinical application. So that's one way to define research. The question about how do you choose what to work on? And I sort of look at it three ways. These are my sort of definitions. What we really are, my laboratory, is we study a specific gene or a pathway or a signaling pathway, we often refer to it as, is just a group of genes that kind of do the same thing. It may tell the cell to move, it may tell the cell to divide at a certain time. So we try to understand that in a lot of detail, and we work on Wnt signaling. And I'll talk about what that is and how basic research really allowed us to understand that over the last 25 to 30 years. You'll hear probably a lot about metabolism, that is how nutrition influences disease, how nutrition influences health because of the new program we have here at the Van Andel Institute in Metabolism. Brian is a member of that program specifically. And then our scientific director, Peter Jones, has spent the last 40 years trying to understand DNA methylation, so how a chemical called a methyl group gets put onto DNA, and that influences human health in the end, because by understanding how this methyl group was put on and doing a lot of work over the last few decades, that led to understanding of how cancer cells modify that particular chemical. And then by understanding that, you can then try to treat cancer with chemicals that interfere with that methylation or change that methylation. So a lot of times the basic research process starts 40 years before and takes that long to apply to the clinic. The challenge that we want to do and sort of our mission is to try to improve the speed of that process overall. So that's really how I would describe our laboratory, is particularly interested in Wnt signaling, and it applies to many different diseases. But other laboratories can come in and say they want to study a specific disease. So you've heard a lot about Parkinson's disease, pancreatic cancer, osteoporosis is what my lab tends to work on more than other things. But that's sort of another way people decide on what to work on, is they want to work on a specific disease. Or you can become an expert. Another way to think about research is you become an expert in tools or methods. So some people with computer science backgrounds and mathematical backgrounds that want to apply it to biology become experts in using mathematic algorithms to understand all this massive amounts of data that's out there. Or our new cryo-EM, there are people that have expertise in doing that specifically as well. And really, I think that the scientific process in a research institute like ours at a university throughout the country works best when you have a mix of people with sort of these interests. So if I work on Wnt signaling in my laboratory, and Wnt signaling is shown to be important in a specific disease, that's often how collaborations happen, is somebody will call me or walk into my office and say, well, I just found that this gene that's related to Wnt signaling is related to this disease I'm working on. Maybe we can talk, and because you're an expert in Wnt and I'm an expert on this disease, maybe we can collaborate and move things forward. So that's sort of how I look at how you define your choice in what to work on. And that's when I say we do work on pancreatic cancer to some degree. We have one person working on the lab, working with Brian in some of the work. But really we're interested in a particular group of genes and how they apply to many different human diseases. If I sort of had to describe our laboratory very simply, we work on Wnt, that's our main focus. But we tend to work on things related to osteoporosis primarily, although we have projects in cancer biology, and then we use genetically engineered mouse models as a tool to study that. And I have background in using genetically engineered mouse models going back about 27 years now. So that's how we've chosen what to work on, is this pathway. So what is this pathway? And I'm not going to go into great detail on it. I'm happy to answer questions if people have interest, but really what the pathway is, very simply, is that there's proteins called Wnt, and I'll get into why it's called that, because it's interesting in terms of how basic research influences development of translation and clinical applications. But Wnt is a protein that sort of floats around outside of cells, and then it binds to what we call a receptor. And when I say bind, this means there's a very specific sort of handshake or lock and key between this, what we call ligand or a protein, and its receptor. So this protein only really binds to this receptor, not the other thousands of proteins that are on the cell surface. That sort of process of physically touching these two proteins together leads to things that happen inside the cell, which we call signaling. In our case, it's a protein that's called beta-catenin, then gets turned on and tells the cell to grow or divide or something like that in the normal case. And this pathway is used in animals as primitive as sponges and hydra, that is, the first multicellular organisms actually use this pathway to become multicellular. So, and then it's been used as animals became more complex in many different things. So almost every tissue in your body needs proper Wnt signaling regulation to form and maybe to maintain itself. So the beginning of Wnt signaling, and I'm just going to present this because of the story of how this came to be is of interest, I think. So almost 30 years ago now, the lab that I trained in as a postdoctoral fellow, at that time in San Francisco, at the University of California, San Francisco, just had a basic question, that is that mice get tumors. So why do mice get tumors? And they found out that a virus caused tumors in mice, and they just wanted to know, how does that virus cause tumors in mice? So what they found was the virus actually inserts itself into the mouse's DNA, and this turns on a gene that causes cancer. They called this gene integration site 1. Integration just means the process of insertion into the DNA. And they called it INT1 for integration site 1. So what they also found about 5 years later was that it was also needed for fruit flies to develop normally. So a lot of times people that don't have backgrounds in basic research often say, well, why do you work on fruit flies? Why do you work on yeast? The reason is that you can understand things very quickly in those systems. You can do things in the matter of days or weeks that would take years to do in mammals or to understand in humans. So there's a big focus on model systems, we call them. We have several laboratories here that have worked on model systems, still do. Heidi Lempradel's laboratory, one of the laboratories in the Metabolism Center, actually works on fruit fly or Drosophila models for those reasons. So what they found, these individuals, when they went and looked to try to understand what this integration gene did that caused mouse tumors, is they found it was the same gene that in the fruit fly causes fruit flies not to have wings when it's missing. So they called it wingless. Fruit fly names are very creative many times. So they called it wingless, and really all Wnt is, that's what we work on in our laboratory, is the combination of this fruit fly gene name with the viral insertion site. They just combined them and called them Wnt1. So Wnt signaling was understood about 30 to 25 to 30 years ago, really developed. Now you go to national meetings on Wnt signaling. We had one, international meetings. We had one in Germany in September. There's one upcoming in Vermont this summer, and there'll be 500 people there, working on things from cancer to osteoporosis to Alzheimer's disease to early development. And so the pathway is used in a lot of different, or applicable to a lot of different things. So what I've really spent the last 23 years working on since I was a postdoc, and this slide is purposely busy because what it shows is it's not just a single protein floating outside the cell, binding to a receptor, turning things on. There's dozens and dozens of genes inside the cell and outside the cell that impact how this pathway works and are necessary for this pathway to work. So really what we work on in the laboratory is these little boxes and circles. One particular postdoc or student may focus on one of these boxes or circles for 5 years. So that's how we advance things on a basic research side. So what types of things do we do? What happens, for instance, when we block Wnt signaling? So I'm going to show you an example of a human patient that is missing one of these receptors, that is, lacks the ability of the Wnt protein to bind and tell the cell to do something. And what happens then is we often block normal growth. There's a lot of examples of this, but this is one example that really we've worked on based on this observation for the last 17 years or so. So this is a disease that's called, very long name, it's called osteoporosis pseudoglioma. It's a very rare disease, but what it is these patients end up often in wheelchairs by the time they're 2 or 3 years old because their bones are so low in bone mass that they just can't walk without fracturing. This patient is a patient of actually one of our longtime collaborators and colleagues, Matt Warman, who runs the pediatric genetic department at Harvard Medical School, Boston Children's Hospital. This patient actually has a blog online talking about their disease. They're missing a gene called LRP5, which is one of these receptors. This patient's actually fractured, I think, about 15 or 16 times in his life. He's actually one of the more mobile patients that are out there. So we study rare diseases like this, not only because we want to help the patients with rare diseases, but it often turns out that patients with these rare diseases, there's maybe 100 of these patients in the world, these rare diseases, if you understand how to treat osteoporosis in these patients, you can then apply it to the thousands and millions of people that have osteoporosis, really because of the same problems. It's just they don't have a genetic mutation present at birth. They develop problems over decades and decades of time. And rare diseases are really underlying most of our understanding of cancer, I would argue, in many cases. We understand colon cancer much better. There's a mutation in a gene in this pathway, actually, in colon cancer that causes people to get colon cancer in their teens and 20s. That's the same gene that gets mutated in almost everybody that gets colon cancer in their 60s, 70s, and 80s. But by looking at these extremes, we can understand things better. In this case, it happens to be relevant to Wnt. And so what we did is we created mice with the same genetic mutation this patient did on the previous slide, and then we examined their bones. These are just x-rays through the sort of hip bone here. So you can see this is a 1-month-old mouse that's lacking these genes. This is a 1-month normal mouse. You can see there's a lot less bone. This is just a little bit farther down their femur, so their leg bone. You can see this mouse is actually already fractured. So what we can do then is use these mice as models. We've done a lot of this work in our laboratory, work with collaborators, work with companies to try to look at, well, can we fix this with therapeutic approaches? So the reciprocal, what happens when Wnt signaling is too high? Well, I kind of gave you this in the intro to what Wnt is. It causes cancer in many cases. This is one of the most commonly mutated gene pathways in cancer. And just some examples, virtually all colon cancer is initiated because of a mutation in this pathway or set of genes. And then almost every other cancer type you look at, there'll be 5, 10, 20% of the cancers will have mutations in this pathway. And that's one of the things we try to do is we call it stratifying patients, but to identify specific genetic modulations or mutations in different types of cancer, and then say, well, if there's pancreatic cancer that has a mutation in this pathway, we should be able to treat it with something that blocks this pathway to turn it off. And my laboratory's also worked on this in parallel with our work in the bone for the last 15 or 20 years as well. So I'll end with a translational application that's actually one of the things that's being translated to is pancreatic adenocarcinoma. So there are several companies that are in what we call phase 1 clinical trials to treat tumors. So phase 1 just means that they're trying to put a new therapy into a patient for the first time and they want to see if it's safe. So you test a few dozen patients and make sure that there aren't problems. Based on actually our publications for the last 10 or 15 years, among other laboratories too, You would have predicted that the side effects perhaps of loss of these treatments would be loss of bone, and that's exactly what the side effect is. So the challenge is that these trials actually worked pretty well in terms of blocking the tumor from growing, but the problem was that large percentage of the patients had loss of bone and started to fracture their bones. So that's obviously not something that's compatible with continuing the treatment. And so what we wanted to do is try to understand, are there ways we can help prevent that loss of bone while still allowing the therapies to work on the tumor cells. So just pointing out the international nature of science, we worked with a team in Singapore over the last 5 years or so to test a way to prevent the bone side effects of cancer treatment. And that's kind of shown here. So what's shown here is we've treated mice for a month like you would a human patient, the same doses. And then we look at x-rays of their bone. And on the side here I've shown sort of a three-dimensional reconstruction of an x-ray of a mouse leg bone. And the part in red here is what we're looking at here in terms of the amount of bone. So if you don't treat the animal, you get this amount of bone. If you treat the animal, it's actually alarming. They lose about 90% of their bone in like 6 weeks if you put them on this. So that's what's happening in the human patients. It's pretty reasonable assumption there. So that's what happens. So they had to stop these trials, not just this one in Singapore, but several other in this country as well. And what we had talked about, because we have some background and expertise in understanding how bone works, because we've worked on bone as a way to apply our knowledge in Wnt signaling for the last 15 or 20 years, is we can treat these mice, we can co-treat them with the cancer drug, but then co-treat them with a drug that blocks bone loss. So this is mice that are co-treated with a drug that many people actually take throughout the country. It's called Fosamax is the name, it's alendronate. So it's a drug from Merck that's used to treat osteoporosis, to block bone loss. So if you now treat these mice both with the cancer drug and the bone loss prevention agent, you get this. So you don't lose bone anymore, you don't get this, and so now you can treat the patients longer, potentially, you can treat the patients more effectively, and that's actually what is going on based on these observations. This group and team in Singapore, David Vership is our collaborator on this. He's at Duke University and the National University of Singapore. He's an MD, or has access to MDs in Singapore that he can work with on trials. They're going in, restarting the phase 1 trial with this approach to see if it works better now on several patients, including a group of pancreatic cancer patients. So just pointing out here, we have collaborators from throughout the world, throughout the country, and we have a lot of collaborators internally here at the Van Andel Institute, people that may work on similar things that provide complementary expertise, and then a lot of people that provide sort of that knowledge in methods and models and microscopy and those sorts of things that become experts in those. So we couldn't do any of this work here without these collaborators. And with that, I think we'll move on to having Brian come up and talk about his work, one of our collaborators in our laboratory. He'll talk about his work really looking at coming up with ways to better detect pancreatic cancer to allow for more informed treatment decisions. So, Brian?
Okay, thanks, Bart. That was a perfect lead into what we're going to talk about with the translational focus. And thank you all for coming and your interest in the Institute and our research. And it's really great to have support from various people in the community and all the people here at the Institute that make this research possible. And I really appreciate the chance to tell you about what we're doing so that hopefully it's understood by more people and can see how basic research then leads to translational research, which can hopefully in the end benefit patients. I'm going to tell you about our work in attempting to detect pancreatic cancer with a simple blood test. And I'm going to tell you about why we have chosen this problem, why it is a challenge, and particularly a challenge in pancreatic cancer, and our approach to dealing with this problem. So there are certain features of cancer that make it really difficult to deal with. One is that it's a disease of the cells, as outlined by Bart nicely, that it arises from one's own cells. This is a particular challenge because the cancer then has many of the properties and functions of normal, healthy cells. It makes it difficult to detect it and to treat cancer in a way that it doesn't affect normal cells. You heard about a severe side effect with one such treatment. So your body is made up of cells of many, many different types, and each cell has its own function. And there are a couple other features of when a cell becomes a cancer cell that make it particularly hard to deal with. I'm going to tell you about a couple of well-known hallmarks of cancer that make it difficult. One is the hallmark of uncontrolled growth. So cells have the ability to propagate themselves, to double in a normal setting, and that's a part of a normal, healthy body, and also when a body is growing, more cells are needed. They produce more cells by the process of division. Certain cells are able to replicate themselves and then divide into two copies of themselves. And that's necessary also in an adult for healing of a wound or replacing dead cells. And so this is a part of a healthy body. But with cancer cells, they hijack this to become uncontrolled. So normal cells, as they grow, they obey signals that tell them to stop growing. But the cancer cells, because of mutations, they no longer obey those signals to tell them to stop growing. For example, when the Wnt signaling pathway is mutated, and they continue to grow. Now, if it were just as simple as that, many cancers could be treated simply by removing the mass. Produces a mass or a tumor. It's usually how cancer is detected. And many benign tumors become quite large and they can be removed, and that deals with the problem. But the second main feature of cancer that makes it much more of a problem is the feature of stepwise progression of severity. So as those cancer cells divide and grow, it makes copies of more cells that also are mutated. But with each time it divides and grows, it can acquire additional mutations that make it more severe, make it more of a problem. It can make them more resistant to any kind of treatment, makes them able to move to other parts of the body. And once it's done that, it makes it very difficult to deal with at that point. The end result of that for a patient then is that the more progressed the cancer is, the longer it's gone and acquiring new mutations and new properties, the less treatable it is. So that really is why it's important to deal with cancer early, and that's what we have chosen to work on, is developing methods to deal with cancer at an earlier stage where it's more treatable. I'm going to tell you about why this is a particular challenge in pancreatic cancer. So a little bit of background on the pancreas to set the stage. The pancreas is an organ with two main functions. It's located beneath the stomach. The first is in digestion. It produces the enzymes necessary for digesting food. And the second is in regulating levels of blood sugar, both relating to nutrient processing. So it secretes molecules into the bloodstream, most notably insulin, but many others that tell the body how much blood sugar to allow in the blood. If the blood sugar becomes too high, it's dangerous, or too low. And the pancreas has a critical role in regulating that amount. And when there's problems, that can lead to diabetes or hypoglycemia. Now, the nature of the pancreas makes it difficult to detect because it is located in the abdomen, situated right beneath the stomach, and it's relatively small. And you can keep that in mind here as I show you what we're trying to do and why this is a challenge. Here's a cutaway view of the pancreas. It's a relatively small organ, but it secretes the digestive enzymes into the intestine, the beginning of the intestine, right after the stomach, so it begins to digest the food. And that it also has the ability then to secrete insulin and many other molecules into the bloodstream for rapid regulation of blood sugar levels. So pancreatic cancer is a big problem. It used to be thought of as a relatively low prevalence disease, relatively rare, but not so anymore. It seems to be increasing as a problem. And this is a projection of cancer death rates from several of the most common cancers in the United States. And up to 2020, this was published about 5 years ago. And at that time, here's pancreatic cancer, was third or fourth. Lung and bronchus cancer, greatly exceeding that, colon cancer number 2. But based on trends in the last 15, 20 years, pancreas has been increasing in death rate, whereas other major cancers have not, or are decreasing. Liver is another one that seems to be increasing. So if this projection continues for the next 10 years or so, pancreas will become the number 2 killer by cancer, surpassed only by lung. This trend probably has a lot to do with the fact that most of the pancreatic cancers are detected late. So 80 to 85%, when the diagnosis is made, these patients are no longer candidates for surgery, which means that the cancer itself has spread beyond the pancreas. They can no longer be treated with surgery. They have to go on to medical treatment. And because pancreatic cancer doesn't respond well to any treatments, chemotherapies, or any known treatments in general, the success rates of those treatments are not very good. So there are many, many people working on medical treatments, drugs for pancreatic cancer, and some of our work may help that other research, but a big recognition in the field is that this number needs to be increased because the earlier they're detected, those patients do better. But the reason that is a challenge, that early detection, is partly because it's an internal organ. There's no way to notice a mass right away. It's not palpable or visible. Also, it's similar to non-cancer conditions, the early symptoms, things like an upset stomach, perhaps, general ill feeling. Also, if there is a suspicion of a problem of the pancreas, the imaging tests are not very good at picking out cancer from non-cancer. Because it is internal, doing a biopsy is very difficult and potentially risky. The pancreas is a very sensitive organ, so whenever it's poked with a biopsy needle, it has the potential for inducing pancreatitis, or if there is a cancer, for spreading cancer. So it's not easy to access, to get samples from. And another a challenge that's been more appreciated recently is diversity between the cancers. So there seems to be multiple subtypes of pancreatic cancer that each have their own features, and so if you develop a way to detect one, that will not detect all of them. So a blood test or a test needs to be able to detect many different subtypes. So in developing a method to detect pancreatic cancer at an early stage, a test, a molecular test, would have to meet these requirements. It has to distinguish the cancer from non-cancer, has to be very specific for cancer, has to find something that is present in the cancer but not present in the non-cancer conditions, so that would be like pancreatitis or other benign conditions. We need to be able to detect the different subtypes, so they can be very different from one another, the pancreatic cancers. And we need to have an accessible sample, so we can't use biopsies for early detection because it's just too hard to get at, or something that requires an endoscopy, for example. Blood is a very good option here because blood is easy to acquire. And because the pancreas is a secretory organ, it seems to often be secreting molecules into the blood, which gives us a real option of working, a real possibility of working. So we have been working on blood tests that meet these requirements here. So how do we approach this problem? I'm going to tell you about the approach that we've taken and some of our results. Well, we began with what's known, and we did have a starting point in this. A molecule that was discovered about 40 years ago, back in 1979. Interestingly, this molecule is secreted by the pancreas is greatly increased in secretion in cancer. And that was discovered, an ability to detect this molecule was discovered about 40 years ago. And it was discovered through a monoclonal antibody. An antibody is a molecule of your immune system that detects specific molecules, and they can be generated for the purpose of detecting specific things. And the name of this molecule, for historic reasons, is called CA19-9. The CA is just as for cancer antigen, and this is just a number designation, just because of the order it was made. When it was originally found, it was not known what it was. It was found that it was up in about half of the patients, more or less depending on how it's measured. If you want to detect cancer without detecting anything else, it only can detect about 20%. Depends where you set the threshold for what you call cancer and what not cancer, but you can say roughly half. And as this test was studied more and more, it turned out that what it detected, which wasn't known at the beginning, was a sugar. A sugar, not like table sugar, but a carbohydrate of which there are many different kinds all throughout the body. Every cell has carbohydrates. Another name for them is oligosaccharides. And another name that's used in the field is a glycan. So this test here turns out that it detects a glycan that's produced and overproduced in many of the pancreatic cancers, not all of them, but a good starting point we felt. We felt, let's begin with this test here and try to understand what's going on here and what's different about the ones that don't produce CA19-9. So that was the beginning premise that we started with. So we asked this simple question: are there other glycans? Glycan is just a term that we use, same thing as sugar, besides CA19-9 that are produced in pancreatic cancer, particularly the pancreatic cancers that don't produce CA19-9. So I said this detects roughly half. Well, maybe many of those that are not detected by CA19-9 are some other sugar, because there are a lot of similarities between the pancreatic cancers. And we realized that if the structure of this oligosaccharide detected by CA19-9 is changed in any way, that test becomes a negative. So what we did was we began by screening through lots and lots of different oligosaccharides that have structures similar to that detected by CA19-9 to see if any of them were good possibilities. Glycans are cell surface molecules that can be very large. Now, what we have here, a little background on glycans, this is from transmission electron microscopy, which is a very, very powerful microscopic method that can allow one to see the inner structures of cells. Now, this is a cell surface from a layer of what's known as epithelial cells. This is a blood cell, also called an erythrocyte. This is an endothelial cell, which means the lining of a blood vessel. And on these linings of cells, the lining of the cell is covered with large, large structures. This is about 1/20th of the whole diameter of the cell, so it extends far out into what's called the lumen, where the blood vessel is, or on the outside of the cell. This provides a barrier and protection. It's proteins that have carbohydrates attached to them. So this is really what protects the surface of the cell, but it also provides the entry point for specific molecules. So all of these have a different structure of the carbohydrate or the glycan. But there are particular structures that are linkage points for proteins and other molecules to attach to. We heard before about Wnt signaling. There's specific attachment points. So there are specific oligosaccharides that provide an identity marker, almost a signature for that cell. So every different type of cell in the body if it's a cell surface like this, or epithelial surface, has a specific carbohydrate code that provides some sort of identity marker for that cell, it seems. And there are then other proteins that can attach to those carbohydrates that allow entry or not entry into the having access to the surface of the cell. So the ability to understand and take apart what those structures are has been very difficult. It's been experimentally difficult. But there's been a lot of progress made in that field over many years, and we've really worked on glycan analysis technologies also. And so I'm going to tell you about the specific structure, a little bit of background in chemistry here that makes up the CA19-9 antigen. And for those of you who have had some chemistry or biochemistry, you'll recognize these depictions here. But what a glycan or carbohydrate is a polymer. So it's made up of units called monosaccharides. And the monosaccharides are then linked together with this oxygen bond here, and they have specific attachment points and linkages which define then the structure of this oligosaccharide. And this is the CA19-9 antigen. So after much research, it was figured out that this is what that test was detecting. So we screened through many different structures, and the one that we landed on turned out to be very similar to the CA19-9 antigen. And you'll notice here that, so this has got 4 monosaccharides. You'll notice this one has only 3. And the difference is that it's missing this monosaccharide called fucose. And it doesn't seem like a big difference, but it actually makes a big difference in what is able to bind to that carbohydrate. And so in the field, we don't really look at these structures very often. We look at simplified cartoon-based simplified structures where we have a different symbol for the different types of monosaccharides. There's many different types of monosaccharides, so we'll just put them together like this so you can easily look at it and know what the structure is with a letter and a number telling you something about the linkage between them. And so this is the shorthand notation for the CA19-9 antigen. It's a tetrasaccharide, meaning it's got 4 monosaccharides, and then this is the one that makes up the STRA antigen. It extends a little farther beyond what's shown here. But you can see it doesn't have that red triangle, it doesn't have that fucose. So what we found then was that this antigen here is produced by many of the cancers that don't produce this one, and that's really the key for detecting a broader range of pancreatic cancers without detecting the benign non-cancer conditions. And so we could show this by simply looking at the tumors themselves. So we had access to the tissue of the tumor, and we asked, are there some tumors that make one and some tumors that make the other? So this is just looking at the actual tissue, the actual cells in the tumors, and we have a way of detecting them on the tissue. This is a piece of the tissue where you can just look at the tissue from the tumor. These are grown in culture, actually, these ones. And then we put a color over where we detected them. So orange is where CA19-9 is, the cyan is where STRA is, and then when they're both there, it's green. You can see that some of them are primarily orange. These are the cancer cells that stain orange. And then on other ones, they're all cyan. So that means there are some tumors that are all STRA, some that are all CA19-9, and then there are some that are both. And there are some that are neither also. And then as we did more research, we found out that the levels in the blood parallel the levels in the tumor. So if the tumor makes primarily CA19-9, the CA19-9 levels in the blood will be elevated. So they secrete into the blood. And that's really the key then for detecting a broader range of cancers. So can we develop a blood test for these types? It really depends upon this relationship that if the tumor cells themselves is a picture of the cell and glycans are on the surface of the cell, this is the CA19-9, this is the STRA, and then do they secrete little proteins or other carriers of that carbohydrate into the blood in relation to what they produce on the cancer cell? So can we use a blood test to detect the type in the tissue? So the steps in discovery and validation, this type of work, there's a lot that goes into it. We have to have great clinical collaborators who are dedicated to research and who work with us, who are very, very good. There's a lot that goes into collecting samples in a rigorous, systematic way, and all the information that goes into it. And then they have to provide the samples to us, and we do the large-scale studies for looking at the relationships then between cancer or subtype of cancer and the presence of particular molecules in the blood. And from that, you can make the initial relationships, initial associations. Once we think we have a relationship, then we specify what we think that relationship is. We fix the hypothesis and we say, okay, let's take some new samples now, and we're not searching for something, now we're testing something. And that's the next step there. We fix a hypothesis and then test it in replicate sets of samples. And then if that validates the hypothesis, meaning so you support the hypothesis, then the next step is to take it to the next level with this kind of translational research that was talked about, where we want to replicate clinical use. And that's what we're trying to do right now, and that would involve, you're not really doing clinical application, but you're replicating it, meaning doctors will collect their sample and send it to a clinical lab and have it analyzed in real time and reported back at the same time frame that an actual test would be used, and then over time build up data asking the question, if we had used this in patient decisions, would it have been valuable? So those are the steps we're taking, and what I'm going to tell you just briefly and show you some results then from these first two of these steps. So we start out after a lot of discovery research where we found that this was a potential new biomarker. We did what we call a training cohort. That's 147 subjects, roughly equally divided between cases, which are people that had cancer, and controls. These are people that had pancreatitis or some other benign condition. This is the level of the antigen in the blood, and each one of these spots is a different patient sample. So this is CA19-9, and this is STRA. And you can see, as a whole, the patients with cancer had higher levels than those without. There's a few that have a little bit of elevation here. And you can see that for the STRA antigen, it's the same. As an individual marker, it works roughly the same. Now, as a standalone marker, as a standalone test, it performs about the same as CA19-9. So it's not a standalone replacement. But the relationship between them makes it very valuable. You can see this here, where this now is a plot where it's taking the same data but plotting them with respect to each other, where so on this axis is the amount of the STRA, and then for the same patient, here's the amount of the CA19-9. So what you see is that there are some patients that are highly elevated in the CA19-9, but not the STRA. And there are other patients that are highly elevated in the STRA, or even down here, but not the STR at the CA19-9. So that means that you can say, if there's a blood sample where it's elevated in either one or the other, then it's likely to be cancer. But if it's elevated in neither one, more likely not to be. So you can bring the two together into a combination test and still have a low rate of false positive detection of non-cancer. So as we then applied this combination test, we compared it to CA19-9 alone. So now this is the performance in percent non-cancer correct. That's how many of the controls did we get correct? How many of the cancers did we get correct? This is the total correct. The cyan is the panel, so the combination test. The orange is the CA19-9 alone. So we set thresholds in what we called cancer, non-cancer relatively high. So 97% of the non-cancers we got right in both, meaning there were 3% false positives. Now, with the CA19-9 alone, we only detected 39% of the cancers. So there were the great majority of the cancers were not detected. But with using the panel, the combination test, we increased that to 72%. So we got somewhere around half of the ones that were not producing CA19-9 with the new combination test. Now that was the first cohort, the training cohort. Then we applied that, we fixed those rules, and we applied it to brand new samples, 50 more, and we didn't change anything about the thresholds or the rules or anything like that. And we again see a great increase. Using these particular thresholds, it was a little lower and higher false positive rate, but we saw a great increase in the number of the cancers that we were able to detect. We can adjust those thresholds to change those numbers a little bit, but we saw a great improvement. Now, the real test of whether something is going to work is what is known as a blinded study, where we receive samples from our collaborators, and we have no idea whether they're from cancer patients or control patients, and they're all mixed up and randomized, and we receive them. And then we run our test here, and we get the assay result, and based on the levels in the blood, we say, we think that one's cancer, we think that one's control, and we do that for a whole set of samples, and then we send our results to what's known as a third party, a biostatistician. We send those off to somebody else, not the doctor who gave it to us. And we say, okay, here's this list of 100 and some samples, and we think that these are the cancers, these are the controls, and then that other person has the result from the doctor and does the analysis and tells us then how well you did, how many % did you get right, so forth. So that's the most rigorous test of whether something is going to be valuable or not. So we did that, and we saw that we see roughly the same thing. We saw roughly the same thing. We saw a great increase in the percentage of cancers that we detected. And this was a really promising result. We didn't get to the 70% like before, but this was a little bit lower too. Every sample set is a little bit different in the composition of the sample set. And also, this is not fully optimized in terms of the assay itself and the thresholds that we use. So this was actually a very encouraging result. This is a statistical term, meaning that we call it statistically significant, meaning there was a less than 5% chance that this was achieved by chance. So this is certainly a real improvement, a non-random improvement. And as we adjusted those thresholds, we became unblinded and we were able to adjust them. We could get the near 70% correct. So we saw this as a validation that we're able to detect a great many more cancers with a very low false positive rate using our combination test as compared to the original. So that was the main thing, the main message I wanted to give you, and I want to just briefly tell you about our current work. We're working with our physician partners and a clinical laboratory to do those clinical studies I told you about, to replicate the actual clinical use. So we are getting our test established in a laboratory where it's gone through all the regulatory requirements, and our clinical collaborators then will send the sample directly to them, and it will be analyzed in real time, and a call will be made whether it was presumed to be cancer or not. And so those clinical studies, there's a lot of work that goes into getting those set up and all the regulations that go into getting things in place for that, but that's coming along. We really are asking good enough for surveillance. Okay, so there's a couple different ways this could be used. One is that a patient has symptoms that are nonspecific, so abdominal pain or something like that, or maybe a mass was detected by abdominal CT, but it's difficult to distinguish cancer from non-cancer. Could be applied to a setting like that, but it also could be applied to a setting where someone is known clinically to have an elevated risk for developing pancreatic cancer. As I said, we don't really screen those patients right now. There's no good way to screen them. they're not at the same level of risk as just anyone out in the population, but they're at an elevated risk whether they have first-degree relatives that had pancreatic cancer, or there are other clinical indicators. New onset diabetes is now recognized to be an elevated risk factor. Other things like that. People like that could, if the blood test works well enough, have an annual blood test to tip off incipient pancreatic cancer that could really help those people. So as we've gone through the numbers, that the cost-benefit analysis, we're getting about to the point where we feel like we would have a chance of being able to use this in a surveillance setting for people that are at an elevated risk for pancreatic cancer. Now, the other question we're asking right now that is very, very interesting as a side note is the patients that make mainly STRA or mainly CA19-9, are those just two meaningless differences between basically the same cancer, or are those two different subtypes, do they behave differently? Do they respond differently to certain drugs, or do they have a different propensity to metastasize? Do they have different clinical outcomes? And in our research so far, we really have seen that there is a difference. The cell culture models, for example, are much more aggressive and resistant. And then the patients that have it in their blood, the STRA, have worse outcomes, many of them. They have shorter survivals. And so we're really working on understanding the differences in the biology between these two types of tumors so that we can understand how they should best be handled and managed. So that we feel is a very fruitful area of research. The other main area that we're working on is to develop additional biomarkers. So this was a glycan, a sugar. We're screening through other glycan structures and sugars to further subtype the disease and develop biomarkers for the subtypes that cannot yet be detected with these two tests that we have right now. So that's what we have going on right now. I appreciate your attention and interest. I want to thank the lab members that have done such a wonderful job in this. These are some former lab members that have contributed, but everybody is contributing to this research really in a substantial way. And as I mentioned, our clinical collaborators, none of this could happen without them, and we've been very blessed with excellent clinical collaborators. I've worked with Dr. Brand at Pittsburgh for many years now, probably 15 years, and other physician scientists who really give a lot of their time and effort to research like this. Oftentimes they do so making less money than they could make without doing research, and so they really do it as a labor of love, many of these physicians. And our core laboratories are very, very supportive here and excellent, and we've been very thankful to receive funding from various sources, federal government and from the Van Andel Institute. So thanks for your attention.
Thank you, Dr. Williams and Dr. Hobb. We do have time for just a couple of questions prior to dismissing for today.
I'm a two-time cancer survivor, breast cancer and fallopian tube cancer. My mother died of ovarian. I have the BRCA2 gene mutation. My great-grandmother died of pancreatic. My mother has two male cousins who've died of pancreatic. So I have the CA19-9. My doctor did that 2 years ago with my physical, and then this year again, or 3 years ago, and then last year. And while it was still in the normal range, it was elevated. I'm not a doctor, so I'm probably not using the right words here, but so I had an endoscopic ultrasound, and I'm scheduled to have another one of those in a couple of weeks just to make sure that everything's normal. Just wondering if you have any thoughts related to any of that, and I'm also interested if there would be opportunity to be involved in clinical trials?
Yeah, I mean, the endoscopic ultrasound can be very good at distinguishing cancer from non-cancer, and so I think that's a great way to go, and with your gastroenterologist can help with that. And as far as clinical trials, we are needing about another year or more to do this next phase I mentioned, where it's really research with just a couple of our partners here. We're working with Mercy Health and Spectrum and a couple others, and we want to build up the data from that, and if everything looks good there, we will start offering the test on a still a limited basis, but it would become available just amongst our partners for more of an advisory capacity rather than directing treatment. So hopefully we can start offering that to patients in the near future.
Dr. Hobb, I'm also at risk for pancreatic cancer, and 1 year I'll have an endoscopy ultrasound, next year MRI. Keep going back and forth. Now, questions are, number one, is it possible to have a normal value of the CA19-9 and increased value of the STRA?
Yes, definitely.
So would that indicate that you are at risk then?
If one had a normal CA19-9, elevated STRA, it could be a risk, or it could actually be incipient cancer. We are trying to figure out, right now we've got some initial research looking at lesions that are still, there's called pancreatic cysts. We think some of the pancreatic cysts, which are precancerous lesions, produce some of this in the blood at low levels, trying to understand whether this can be used as understanding a risk for development of full pancreatic cancer. But it also, if it were positive, could indicate that there already is something there.
Okay. And number two, do you have any idea at what stage in the development of the pancreatic cancer that these values would begin rising?
Well, we know that at the still resectable early stage, stage 1, stage 2, they already are detectable in the blood, although I would say the percentage of patients that have the detectable levels is lower for that group than those who are at the late stage cancer. So we think that for the very early that it's harder to detect clearly. And probably a smaller percentage of patients are detectable. But it's hard to get data on that because it's hard to know exactly what stage that cancer is just from the blood samples.
Okay,
So,
thank you.
yeah, sure.
So Brian, my question is about, in the middle, the question is about your false positives. What gold standard test are you comparing to call those false positives with your double test? And really, how sure are you that they are false, right? Rather than that early, difficult to detect cancer, or that there's some other interesting story in there?
As I have gotten into this research and worked directly with our clinical partners at Mercy and Spectrum, and you realize just how uncertain this is sometimes, where you sometimes hear a diagnosis and you think it's just black and white, it's this or that, and you know that's kind of comforting to the patient sometimes, but in reality it's based on a lot of circumstantial evidence, and in the end a decision has to be made, and you have to go with what you think is most likely based on all the evidence, but we're struggling with this too. I mean, we see false positives, and we've talked with our doctors, and he said, "Wow, well, I wonder if I should go back and take a more careful look at that patient." You know, so I think in most of the time the correct diagnosis has been made, but I think there are some cases where not. So, but you hit the nail on the head with that. It's a challenge.
We need to wrap our conversation up today. We want to thank you once again for joining us for the lecture series. As a global leader in biomedical research, we feel like it's our community responsibility to bring the science that we're doing here at the Van Andel Institute to you, our community members as well. And so we're grateful for your willingness to participate with us in this capacity. We're also grateful for the Van Andel family who both helped us build the facility as well as help provide some of the undergirding for the research work that we do. As you can see through the slides and the presentations today, we also receive funding from various sources that include the National Institute of Health, Department of Defense, and many other organizations, but we are still dependent upon philanthropic support that helps some of these projects in its early stages to help launch off the ground. And so we're really dependent on the entire community for us to do the work that we're doing here in biomedical research. Want to thank you once again. We will have additional lecture series throughout the year. We'll have 3 additional ones. You can watch our website, provide us with your email address, and we can communicate with you that way. But thank you once again for joining us today.